Greetings everyone. So, in the course of the plant physiology and metabolism and in continuation to lipid biosynthesis, so we have another role of the lipid, that is lipid catabolism. So, in today's lecture, we will delve into the intricate process of the lipid catabolism, that is the fundamental aspects of cellular metabolism with significant implication for energy production and homeostasis. Lipids, comprising a diverse array of molecules such as fats, oils, and phospholipids, serve as a vital energy stores and structural components within cells. Understanding the mechanism underlying lipid catabolism is essential for comprehending how organisms, especially plants, derive energy from these molecules and maintain metabolic balance. Lipid catabolism primarily involves the breakdown of the complex lipid molecules into simpler components, ultimately yielding energy in the form of ATP. So, the main pathway through which lipids are catabolized is the beta-oxidation, that is a series of enzymatic reactions that occur within cellular organelles such as mitochondria and peroxisomes. Just have a look on the introduction. In oil seeds and pollen grains, lipids constitute a significant proportion of the dry weight, often reaching to 60%. Triglycerides, the primary storage form of lipids, are housed within the endosperm of cotyledons of oil seeds. During the germination process, which initiates the seedling growth, these triglycerides undergoes hydrolysis, that is mediated by the enzyme known as lipases. So, this hydrolysis results into the breakdown of triglycerides into glycerol and fatty acids. So, lipases exert their action at the interface between oil bodies and water, facilitated by their binding to oleosin, that is the structural protein which is present in all the oil bodies. In the context of castor beans, a well-studied example, that is the two types of lipases are identified, that is acid lipases and alkaline lipases. Acid lipases are capable of acting on mono, di or triglyceride and are localized within oil bodies. Remarkably, these lipases maintain their activity even in the dry seeds. Conversely, the alkaline lipases are situated within the membranes of the peroxisomes and exhibit activity only upon the absorption of water by the seeds. They specialize in the hydrolysis of monoglycerides. This intricate interplay between lipid storage, hydrolysis, and enzyme activity is fundamental to the energy and nutrient provisioning required for successful seed germination and subsequent plant growth. In plant metabolism, glycerol and fatty acids follow distinct pathway. Glycerol is processed within the cell cytosol, whereas fatty acids undergo transportation to peroxisome, particularly specialized ones called glyoxysomes. It is found in non-green parts of the plant. Once in the cytosol, glycerol is enzymatically converted into dihydroxyacetone phosphate through the action of glycerol kinase and FAD-linked glycerol 3 phosphate dehydrogenase. So, dihydroxyacetone phosphate is then further metabolized via glycolysis, contributing to energy production and carbon metabolism within the cell. Meanwhile, fatty acids are shuttled to peroxisome through the action of an ATP binding cassette, that is transporter, known as the camatose CTS, which resides in the peroxisomal membrane. Within the peroxisome, fatty acyl groups are activated by long chain fatty acyl-CoA synthetases, particularly LACS6 and LACS7, forming fatty acyl-CoA molecules. This activation process necessitates the presence of ATP and coenzyme A. During seed germination, fats undergo conversion into sugars, a critical process for providing energy and building blocks for growing seeds. This conversion entails several steps, including the beta-oxidation of fatty acid, the glyoxylate cycle, and gluconeogenesis. So, beta-oxidation occurs in both peroxisomes and mitochondria, generating acetyl-CoA molecule. The glyoxylate cycle, uniquely found in glyoxysomes, allow for the transformation of acetyl-CoA derived from fatty acid degradation into carbohydrates. Whereas the gluconeogenesis then takes place in the cytosol. These newly synthesized carbohydrates are formed from non-carbohydrate precursors, including intermediate from the glyoxylate cycle. Additionally, during the plant senescence, the breakdown of the membrane lipids occur, albeit at reduced rates compared to germination. Unnecessary fatty acids are recycled and the resulting carbohydrates are transported to support other parts of the plant. Notably, the absence of glyoxylate cycle in animals prevent the conversion of fats into sugars in animal metabolism, highlighting a distinct metabolic divergence between plants and animals. As you can see here in the figure, the cytosol, glyoxysomes and mitochondria are participates in the lipid catabolism. As we have discussed in the previous slide in plant metabolism, that the glycerol is converted to dihydroxyacetone phosphate in the cytosol, while fatty acids are transported to peroxisomes, specifically glyoxysomes in non-green parts. Glycerol is processed through glycolysis, whereas fatty acids are activated to fatty acyl-CoA within peroxisome. During the seed germination, fats are converted to sugars via beta-oxidation, the glyoxylate cycle, and gluconeogenesis. Beta-oxidation yields acetyl-CoA, further process in the glyoxylate cycle to form carbohydrates with gluconeogenesis occurring in the cytosol. The senescence triggers membrane lipid breakdown with the recycled fatty acids supporting carbohydrate synthesis. So, beta oxidation is a fundamental metabolic pathway involved in the degradation of fatty acid that is characterized by the sequential removal of two carbon fragments in the form of acetyl-CoA molecule. So, this slide will discuss about the beta oxidation of the fatty acyl-CoA. This process occurs in both the plants and animals, albeit with some differences in their cellular localization. In plant cells, beta oxidation takes place within specialized organelles called the glyoxysomes, whereas in animal cells, the mitochondria. Glyoxysomes are particularly abundant in the endosperm cells during the germination of acyl seeds. The process of beta oxidation comprises four main enzymatic steps, and the first one is the acyl-CoA oxidase, which catalyzes the dehydrogenation of fatty acyl-CoA at the 2 and 3 carbon atoms, forming a trans delta 2 acyl CoA molecule. Subsequently, acyl CoA hydratase facilitates the hydration of trans delta 2 acyl CoA, resulting in the formation of hydroxyacyl CoA. Following this, beta hydroxyl acyl CoA dehydrogenase mediates the oxidation of the hydroxyacyl CoA, leading to the synthesis of ketone beta ketoacyl CoA. Finally, acyl CoA acetyl transferase, that is thiolase, catalyzes the thiolytic cleavage of beta ketoacyl CoA by coenzyme A, yielding acetyl-CoA and a fatty acyl-CoA shortened by 2 carbons. This series of reactions forms a cyclic process that repeats until the entire fatty acyl CoA is fully degraded. The number of cycles required for complete degradation depends on the initial length of the fatty acids hydrocarbon chain. For instance, a 16 carbon palmitoyl CoA molecule necessitates 7 cycles of beta oxidation to produce the 8 molecules of acetyl CoA. This process is essential for energy production and carbon metabolism in plants, providing a crucial mechanism for utilizing stored fatty acids during seed germination as well as other growth processes. As you can see in the figure that the beta oxidation of the fatty acids, as we have discussed in the previous slide, that the beta oxidation is a metabolic pathway which is essential for the fatty acid degradation, involving sequential removal of two carbon fragments as acetyl-CoA. While occurring in the glyoxysomes in plants and mitochondria in animals, glyoxysomes are prevalent during seed germination. This process comprises four steps, that is the dehydrogenation, hydration oxidation, and thiolytic cleavage, that is catalyzed by the specific enzyme. Acetyl-CoA is generated cyclically until full degradation, with the number of cycles dictated by the fatty acid chain length. And this pathway is pivotal for energy and carbon metabolism in plants, particularly during the germination, enabling efficient utilization of stored fatty acids. In plants, the structural organization of enzymes involved in the beta oxidation presents a notable departure from that observed in the animals. Unlike in animals, where each step of beta oxidation is typically catalyzed by distinct polypeptides, plants feature a unique arrangement where enzymes responsible for the second and third steps of the pathway are integrated into separate domains of multifunctional protein. This consolidation of enzymatic function within the multifunctional protein streamlines the process and allows for efficient coordination of beta oxidation. Furthermore, the multifunctional protein encompasses auxiliary activities essential for the oxidation of unsaturated fatty acids such as D3-hydroxyacyl-CoA epimerase and delta-3 delta-2 enoyl isomerase, facilitating the handling of these substrates during fatty acid degradation. During this integration, enzymes governing the first and fourth step of the beta oxidation in plants remains as a separate polypeptides. This modular organization likely confers versatility and fine-tuning capabilities to the beta oxidation processes in the plants, ensuring optimal utilization of fatty acid that is reserved during various physiological processes, including seed germination and energy metabolism. Overall, this distinct structural arrangement underscores the complexity and adaptation of plant metabolism to efficiently cope with, with diverse environmental and developmental cues. As you can see here in the figure, in plants, the enzymes responsible for beta oxidation are organized into a complex that is known as the multifunctional protein. So, this complex comprises several enzymatic components, each playing a distinct role in the sequential breakdown of the fatty acids. Among these components are acyl-CoA dehydrogenase, that is denoted by the E1, then acyl-CoA hydratase, that is denoted by the E2, L-beta hydroxyacyl-CoA dehydrogenase, denoted by the E3, and thiolase, denoted by the E4. So, notably E1 and E4 exist as separate polypeptides within the complex. However, the E2 and E3 along with two additional enzymes involved in the oxidation of unsaturated fatty acids, that is namely the D3-hydroxyacyl-CoA epimerase, which is denoted by the E5, and delta 3 delta 2 acyl-CoA isomerase, denoted by the E6, are integrated as separate domains within same polypeptides. This structural arrangement optimizes the efficiency of the beta oxidation process by facilitating the coordinated interaction and sequential catalytic activities of the enzymatic components within the whole MFP complex, that is a multifunctional protein complex. So, in contrast to animals, where enzymes involved in the beta oxidation exist as a separate polypeptides, plants exhibit a distinct organizational strategy. Specifically, enzymes responsible for catalyzing the second and third steps of beta oxidation are integrated into a separate domain of a multifunctional protein. This structural arrangement within the multifunctional protein streamlines the beta oxidation process, enhancing coordination and efficiency. Additionally, the multifunctional protein encompasses auxiliary activities necessary for the oxidation of unsaturated fatty acids such as D3-hydroxyacyl-CoA epimerase and delta 3 delta 2 acyl-CoA isomerase within its other domain. These auxiliary activities play a crucial role in handling unsaturated fatty acid substrate during the degradation process. Meanwhile, enzymes governing the first and fourth step of beta oxidation remain as separate polypeptides in plant. This separation ensures the specificity and effectiveness of each enzymatic functions. Overall, the molecular structure of the MFP provides a level of flexibility allowing for the accommodation of various substrate and metabolic demands within the plant cell, while simultaneously optimizing the beta oxidation pathway for efficient fatty acid degradation. Moreover, the beta oxidation, while primarily occurring in the mitochondria in animals, also takes place in the peroxisomes. In the initial oxidation step of beta oxidation occurring within the peroxisomes, the process differs significantly from that in mitochondria. Here, acyl-CoA oxidase, a flavin-linked oxidase, plays a pivotal role by directly transferring 2 electrons to oxygen. This transfer results in the production of hydrogen and the liberation of energy in the form of heat. Unlike in mitochondria, where the first oxidation step leads to synthesis of ATP through the electron transport chain, the energy released in peroxisomes is dissipated as heat. However, the generation of the H2O2 poses a potential risk as it is a toxic byproduct. To mitigate this toxicity, peroxisome contains 2 enzyme systems. One such enzyme catalase is soluble and acts promptly to degrade H2O2 into harmless water and oxygen. This rapid detoxification mechanism ensures the maintenance of cellular homeostasis and prevents oxidative damage within peroxisome or glyoxysomes. Another mechanism employed for detoxifying hydrogen peroxide within peroxisomes and glyoxysomes entails the utilization of membrane associated enzymes. Specifically, this process involves the action of 2 key enzymes, namely ascorbate peroxidase and NADH dehydroascorbate reductase. Ascorbate peroxidase, an enzyme located within the membrane of peroxisomes and glyoxysomes, play a crucial role in the detoxification of H2O2. This enzyme utilizes ascorbate, that is vitamin C, as a cofactor to catalyze the reduction of hydrogen peroxide to water, thereby mitigating its harmful effects within the cellular environment. Ascorbate peroxidase achieves this by utilizing the reducing power of ascorbate to neutralize H2O2, generating monodehydroascorbate as a byproduct in the process. Alongside NADH dehydroascorbate reductase, that is the another membrane associated enzymes, participate in the regeneration of ascorbate to sustain the activity of ascorbate peroxidase. NADH dehydroascorbate reductase catalyzes the reduction of monodehydroascorbate back to the ascorbate, which utilizing NADH as a cofactor. This enzymatic reaction ensures the continuous availability of ascorbate, which is essential for the sustained functioning of ascorbate peroxidase in detoxifying hydrogen peroxide. Together, these membrane associated enzymes, ascorbate peroxidase and NADH dehydroascorbate reductase, form a crucial detoxification system within peroxisomes and glyoxysomes, effectively neutralizing hydrogen peroxide and maintaining cellular redox homeostasis. In the second oxidation step of beta oxidation, The enzyme beta-hydroxyacyl-CoA dehydrogenase facilitate the dehydrogenation of beta-hydroxyacyl-CoA. This process results in the reduction of NAD to NADH, producing a reduced form of nicotinamide adenine dinucleotide. However, in peroxisomes or glyoxysome, where beta oxidation occurs, there is no electron transport chain as found in the mitochondria to oxidize NADH. Instead, NADH oxidation is coupled with the reduction of monodehydroascorbate to form vitamin C, that is catalyzed by the enzyme MDA reductase, that is MDAR. Additionally, malate dehydrogenase, that is MDH, within peroxisomes also participate in NADH oxidation. MDH catalyzes the reduction of the oxaloacetate to malate, with malate being transported to the cytosol for oxidation back to oxaloacetate to NADH. This oxidation is coupled with the reduction of NAD to NADH. Notably, the reactions involved in beta oxidation of fatty acids in peroxisome, glyoxysome bear resemblance to the reversible reactions observed during the synthesis of fatty acids, highlighting the interconnectedness of fatty acid metabolism and cellular processes. The biosynthesis and beta oxidation are two fundamental processes that is involved in the metabolism of fatty acids and occurring in both the plant as well as that of the animal. However, there are some key differences in how these processes operate in plants compared to animals. This table shows a comparison between the biosynthesis of beta oxidation of fatty acids in plants. Fatty acid biosynthesis and beta oxidation are two essential processes involved in the metabolism of fatty acids, each occurring in distinct cellular locations and characterized by a specific enzymatic activities and reaction mechanism. In terms of cellular location, fatty acids biosynthesis predominantly takes place in the plastids, particularly in the plastid envelope and chloroplast stroma in plants, whereas beta oxidation occurs in glyoxysomes in plant cells. The activation of fatty acids differs between these processes, with fatty acid being activated as thiosters of acyl carrier protein during the biosynthesis and as thiosters of coenzyme A during the beta oxidation. Regarding the size of fatty acid, biosynthesis in the plants typically involves the synthesis of fatty acid up to 18 carbon atoms in length within plastids, with further elongation potentially occurring in the endoplasmic reticulum. In contrast, beta oxidation in glyoxysomes can degrade fatty acids in all sizes. The enzymes involved in fatty acid biosynthesis include the fatty acid synthase, which consists of several enzymes in addition to acetyl-CoA carboxylase. On the other hand, beta oxidation requires a complex of 4 enzymes to catalyze the breakdown of fatty acids. Redox reaction cofactors play a crucial role in both processes, with fatty acid biosynthesis that requiring NADPH as a reducing agent, while beta oxidation involves flavoprotein dehydrogenases that directly transfer electrons to oxygen producing NADH. The processes themselves differ fundamentally. Fatty acid biosynthesis involves the elongation of fatty acid using malonyl ACP as a 2-carbon donor, while beta-oxidation entails the sequential removal of 2-carbon fragments as acetyl-CoA molecules. Furthermore, the beta-hydroxyacyl intermediate in beta-oxidation exists in the D-isomer form, whereas during biosynthesis, the intermediate typically exists in the L-iso form. In summary, while both fatty acid biosynthesis and beta-oxidation are essential for lipid metabolism, they exhibit distinct characteristics in terms of cellular location, activation, fatty acid sizes, enzymes involved, redox reaction processes mechanism, and intermediate forms. These differences reflect the diverse metabolic needs and adaptation within cellular compartments in the plants. So, this is all about the today's lecture. This lipid catabolism will continue in the coming lecture. Thank you.